on the Planteome project here at Oregon State University with Pankaj Jaiswal. We're a fairly new project which grew out of the plant ontology project. So we were funded in early in 2015. So we're just starting our second year here. And we appreciate the funding from NSF, of course. And I put the web link on here if anyone wants to go and check us out online. If we have time, I have slides today, but if we had time at the end, I could give a little live demo. I didn't know how the time would go, but so for the meantime, I thought we would go through some slides and just give you an overview of some of the things we're doing. So the Planteome is a web-based resource and a database for reference ontologies and applications for plant biology. The vision of the Planteome project is a centralized online platform where ontologies, both reference ontologies and application ontologies, which I'll explain a little bit more about as we move ahead, for all different kinds of plants are used to annotate a wide variety of data types. And I put some examples on the slide here, which you can see, such as traits, phenotypes, plant diseases, of course, anatomy and development, which was our area of expertise in the plant ontology project. We also have a lot of information on gene function. We are some new information with germplasm and germplasm sources. And all these are related back to the treatments and environments from the experiments they were created in. And we also have data concerning mutant phenotypes and QTLs. So on the lower part of the screen, I just have an example of some different traits that we might be describing in the Planteome for such things as color, shape, and size. And you can see these three different types of plant materials are very different in their anatomy. We have an example of cassava storage root, soybean seeds with different colored seeds and different patterns on them, and then potato tubers, which have different colors, different internal characteristics as well, different colored skin and different colored flesh. So it's challenging to cover all these different types of data, and the scope of the Planteome is quite broad. Was there a question, Lisa? Oh, sorry. I didn't know. I thought someone was asking a question.
There was an echo, so if you're not speaking, please mute yourself.
Yeah. So the scope of the Planteome is quite broad, and it ranges from the very high-level areas such as taxonomy, the environment and climate, and then you can see these tracks through this schema show you different types of data that we host and how we annotate it. So we have germplasm, which is represented by stalks and accessions, and we have traits in that germplasm. We also have genetically characterized polymorphisms in that germplasm, which has been related to traits. And these are expressed in terms of the transcripts that are formed, the peptides that are produced. And this is a big interactive environment, of course. Genes are expressed, in pathways. There's synteny between different genes, which are mapped to genomes, the loci in the accessions, and so on. So this just wanted to give you kind of an overview. And on the right-hand side here, I can show you there's a number of different ontologies listed, and the bottom two in green are the plant ontology, which covers growth stages and plant structures. And so any of these things like the genes could be expressed at these different stages in these different structures. We also deal with gene ontology annotations, and so there's the three aspects of the gene ontology: molecular function, biological processes, and cellular components. And the overarching idea of this is to capture phenotypes, which we use the plant trait ontology captures, and it encompasses all of these different aspects. So, hang on one second, my slide is not advancing. Try another way here. There we go. So I thought some of the people on the call may be familiar with what ontologies are, but I know that this is a very diverse group. So I thought it would be helpful maybe to just give you a very short introduction about this thing that you hear about often, probably nowadays, what is an ontology. So an ontology is really defined as a formal conceptualization of a specified domain of knowledge. So it's a way to describe things that exist in reality. And the key features of ontologies are that the terms are defined, so the names of the objects are defined, and their relationships between the terms are defined, which is very important. And the relationships are shown in this diagram below by the blue lines and the orange lines. And there's different kinds of relationships in different ontologies. These are two of the probably the most common relationships: the ISA or subclass relationship and the part of class or subclass relationship. So you can see in this example from the plant ontology, and this is the anatomy branch, or the plant anatomical entity branch of the plant ontology, you can see a plant structure is a subclass or is a plant anatomical entity. And these relationships give the ontology their power, and they allow logical inference by computers across the ontology. And through these relationships, the terms are arranged in a hierarchy. You can see kind of a graphic here showing a very small part of the plant ontology hierarchy. And in order to build these ontologies, we use different languages or knowledge representation languages, which are represented here by these acronyms RDF, OBO, and OWL. RDF is a resource description framework. And OBO, I was just thinking this morning that I'm not exactly sure what OBO means. I think it's the ontologies for biomedical. This is, oops, I'm sorry, I went too far there. But OBO is like a version of the OWL, which is an ontology web language.
It's open biomedical.
Oh, open biomedical ontologies, of course. And the OBO is more of a textual based, where OWL is more XML based, I believe you would say. So if you look at the spectrum of different types of ontologies, there's really a range of semantic clarity or semantics, and a range of how much it costs you and how hard it is to make them. So the simplest glossaries you're familiar with, like what you would find at the end of a textbook, and they go up in complexity through controlled vocabularies, data models that might be used by different companies, taxonomies, and the top level is really ontologies. They have the most semantic strength or highest semantic clarity, and they're the most work to make and the most expensive to make. And so you can see the range of different languages on the lower axis here. So I have to acknowledge this is not my slide. This is a slide that goes around, and it's originally from this link here. So this is another graphic showing the Planteome suite of reference ontologies that we have, and this shows another way to show you the range of granularity, ranging from terms in the environment, describing the environment that the plant lives in, down to individual molecules. And each of these are different ontologies. The yellow ones are ones that we develop here in-house for the Planteome project. That's the plant ontology, the plant trait ontology. This is the other aspect of the plant ontology. And we also have a plant environmental conditions ontology. And we have a plant stress ontology, which is currently under development, which spans a wider range of this granularity spectrum. And along the way, we have, in the green, our collaborator ontologies who we work with. One of the really important one of the really important aspects of working with ontologies is we always try never to overlap with what other people are working on. So it's very collaborative. The ontologies such as the GO and the phenotypic quality ontology, PATO, we integrate them and use those terms to describe the aspects of our data which we need the quality for, or biological process or molecular function. We also use a lot of terms from the chemical entities for biological interest, CHEBI, the protein ontology, and also, of course, things are related back to their meaning in the NCBI taxonomy. So, As I mentioned before, the Planteome ontologies are linked to data from many, many species, and I'll give you some numbers about that in a moment. But this is just one of our examples that we like to show people of some of this diversity across plant species when you're looking at fruit color traits. So all of these pictures show different fruit colors in different crops. This is wheat and corn. The kernels are actually, this caryopsis or the kernel are actually a fruit. The banana fruit, tomatoes, which are actually a berry, soybean pods. And this is a tricky one. This is one we always throw in to throw people. The fruit on the strawberry is actually the little yellow akene on the surface, not the big red fruit, which you like to buy in the supermarket or get in your garden. So with all these different traits, these are all related back to the data which is behind them, has been characterized, the genes. There may be mutant genes, there may be QTL studies that have been done, and of course there's germplasm that has been characterized for these. And this is related to all these different domains such as phenotypes, gene expression, function, the domains and the interactors of the proteins. And so an important thing to remember is that in the Planteome and in most ontologies such as the gene ontology, there's the ontology terms, there's the data, and in between those are the annotations, which are links between the ontology terms and the data objects. And that's what gives the database the power, because you have the relationship between the ontology terms and that hierarchy and the data that exists in there. So we're very excited. We just released our first full release. We have had kind of a beta release out for several months this year. Our first full release just came out in the beginning of September. And we have all the reference ontologies that were listed on the previous slide, as I mentioned before. And in addition to that, we've integrated currently 4 species-specific crop ontologies from the Biodiversity Project. And we've been working very closely with that group for quite some time. So currently we have traits from cassava, rice, lentil, and wheat. And we have 2 more that are very close to being ready to be integrated. I think it's maize and sweet potato. And in our database, we link out to the data providers for all these different crops and traits, which I'll show you an example of in a moment. So currently we have about little over 17 million annotations, which remember are the links between the ontology terms and the data objects. This covers about 86 different plant taxa, which we've been pulling in from about 24, 25 or 6 database sources. This changes every few days, so it's hard to keep track of it. But in the up-to-date release that we have out right now, there's about 24 different database sources. And I know a lot of you, as we went through the introductions, are from groups who we work very closely with, so you're probably familiar with that. We have about almost 2 million data objects in the database. Many of these are genes and gene products. We also have a large number of germplasm sources and also QTLs that have been characterized in the different crops. One of the exciting features of the Planteome, which is new for our project, is we have functional Go annotations for about roughly 62 plant taxa. And this has been done in-house using a customized in-paranoid analysis of over 100 species across the tree of life. And this is performed using a gene family clustering based on orthology, and then an interpro scan for de novo Go annotations as well. And in our flowering plant species in the Planteome, those have been enriched based on orthology to Arabidopsis, which was characterized by tear and Go. The Go annotations were. So this is a really unique resource at the Planteome because many of these crops or many of these plants are not well studied and not well characterized. So it may be the only place for many of these plants that you can come and find those Go annotations. So yeah. That's kind of exciting information to share with you. So as I mentioned before, we're on the web. We have a Planteome browser at planteome.org. And at the Planteome browser, you can do a number of different things. You can search for terms and data there, and you can, this is the new feature that we have in our release is this drill down browser. And this is actually a hierarchy of the ontology terms. And you can click on the little plus buttons and it will expand. And in the gray box or the gray circle shows you the number of annotations to that term. So as you drill down through the ontology, you can click on that gray box and go and see what genes or QTLs or germplasm is annotated to that term. So on the web page, you can see that anything that's in blue is a hyperlink. So this is showing you if you went to this fungal disease resistance, you could drill down through the ontology and find out where that was. You can also link out, as I mentioned, to our data providers. This link here from the Hordeum vulgare barley name links out to the NCBI page. And of course, we have a lot of maize GDB data, and we have links out from those. You can't see it, it's hidden behind this guy, but this is a gene model which is involved with Helminthosporium carbonium susceptibility, or this is a gene for HM2, yeah. And so you can link out to the source of the data to get more information. And another example is all of the information in the database is referenced. Some of it goes to PubMed and some of it goes to specific database references. So this shows an example of a link out to Gramines, a reference at Gramine for this gene that you're looking at. And another example here is from the Soil Genomics Network. We have a locus for Fusarium wilt race 2. So feel free to come and explore and get familiar with what you can find at Planteome. There's lots of information there. So I wanted to talk a little bit more about the species-specific ontologies and the data that we're importing from them. As I mentioned, we've been working closely with Bioversity, and I have 3 examples here from the CG centers which have been integrated into the Planteome. The first one is cassava. We had a workshop here in Corvallis last fall, and we had several representatives from some of the CG centers visit. And the representative from IITA came, and for each of these crops, they have very specific traits that are very useful for the plant breeders. And in order to integrate those, we have to map between the traits from the application ontology to the reference ontology. So in cassava, we have about 175 traits, mostly to do with the storage route, but also disease disease susceptibility or severity, and this one is a red spider mite, which is an insect, these examples that are shown here. We had a curator come from Acarta who works on lentil, and so we have a selection of lentil traits as well. And as well, we have a fairly large number of traits from the Erie rice breeding collection, which are specific to rice. So the question is, how can you make interspecific comparisons across these ontologies? How can you find data from these different ontologies if they're all out at their different CG centers? And that's the power of integrating them with the reference ontology. So this schema shows, this is an example of plant height, which is a fairly common trait across different plants. And so we have 4 examples here of traits from the crop ontology of lentil, rice, wheat, and cassava. And so the annotations that are attached to these terms will flow up through the relationships in the ontology to the reference ontology term for plant height. So the black lines here are the ISA or subclass relationships, and then the blue lines are what we call adheres in relationships. For example, the pedo terms which describe the quality. So this is for plant height. Pedo height adheres in the whole plant, and that is the way that the plant height term is constructed in the TO. and it allows us to navigate through the different ontologies with these interrelationships. So this is a very collaborative project, working with these different groups. And this is just another schema showing in a graphical sense how those three ontologies interact with the reference trait ontologies. So these are the three crop ontology examples that I showed you before. And this could be the disease traits, or it could be plant height, or any of the traits that are in common. But by mapping back to the reference trait ontology, we allow researchers to come in and ask, what traits are expressed in plant height in these various plant species. So one thing I wanted to talk about briefly was our new data annotation pipeline. This is some work that Austin Meyer has been doing in our lab. We've been collecting germplasm annotations from Green Global using the Samara tool. You can see the nice little graphic they have for the Samara here. So the germplasm data is scraped or collected from the web using the Samara tool, and it's mapped to the descriptors in the trait ontology, which has to be done partially manually, and then that's converted into our GAF2 format to go into our database. And Austin has worked hard at developing some nice scripts for doing this, which is really great. And so the GRIN database has about 6.6 million observations for about 5,500 unique GRIN taxa. So there's a wealth of information out there that, by annotating it with the ontology terms, makes it more accessible for people to use and share. So this is just another example. Once the data is integrated into the Planteome, how you can access it. This is an example of a lentil germplasm called Red Chief. And this is the data page on our website. So you can see this has been sorted by the bioentity from GRIN. So there's links from this page out to the original source provider, of course. And also on this page, you can download selected data using the download button. And as I mentioned, this is a link out here, which takes you to GRIN. Yes. So we're integrating links between these different resources and making the data more accessible. So I wanted to just mention a couple of other things that we're doing. We are developing some practical tools for ontology development and collaboration. All of our ontologies are available on the GitHub site. Many of you may be familiar with GitHub. We encourage people to come here and visit us, and you can request terms and you can make comments. This is a great tool for sharing and tracking revisions. There's an issue tracker for each of the ontologies where you can suggest changes. If you see something that's missing, please let us know, or if you see something that you think needs to be modified. We're also developing application program APIs, application program interfaces, to access the Planteome ontologies from any apps that you might be interested in creating. And right now we have 2 different APIs. We have a fast search, which just returns the search term and the ontology. And so that's kind of a fast one that you can use in search boxes. And there's also one with detailed term data. So these are currently under development, and they're being filled out. But an important thing to know is that all of the reference ontologies and the integrated application ontologies are available through these APIs. And if you're interested in integrating this into your work, please just contact us and let us know. Another thing that's being developed at Planteome in collaboration with University of California at Santa Barbara and Cyverse is a BISC web-based image segmentation and ontology annotation for images. And this is linked to the APIs. So when you select an area such as this shown here with the petal, you can start typing in petal and it will pull in the ontology information, and that will be stored in the metadata for that image. So it's a very important part of the Planteome that's being developed because, as we all know, images are a big deal, and that's how a lot of people communicate things nowadays. So how can other groups get involved, and how can they contribute and utilize these resources, especially for the plant people, but not just for the plant people, because we carry a lot of this Go information, which can be valuable if you're doing analyses, but we encourage people to come and explore the annotations and ontologies for your own work. Please, if you're interested, create a GitHub account if you don't have one, to request terms and make comments or share your own ontology if you're building one. And we'd love it if you wanted to annotate your data or have us help you annotate your data and submit it to our repository. If you'd like to have it integrated into the Planteome, we can definitely talk about that. If you're interested, you might be interested in coming to an upcoming workshop for some hands-on training. This is a photo from last fall's workshop where we had these folks come from all over the world and spend about 4 or 5 days in Corvallis, where they learned about ontology theory and practical tools, and we did quite a bit of hands-on work with the ontologies and data annotation and mappings. So some of the folks that attended were the CG curators, the 3 CG curators from ICARTA, IITA, and International Center for Potato Research in Peru. And in addition, our friend Rex Nelson, who's on the call today, was a member, and he was representing Soybase. So we had a wide range of people, in addition to all the Planteome people and ontology people who were there. So who all is involved? This is a big project. There's lots of groups involved. The project's led here at Oregon State University by Pankaj Jaiswal, and we have 2 computer science co-PIs at Oregon State here are also involved. Chris Mungle from Lawrence Berkeley National Lab is a co-PI. Elizabeth Arnaud at Bioversity in Montpellier, France, with Marie-Angelique, and as I mentioned before, some of our CG curators, and there's many, many of them. And we have a few other people. Barry Smith is a consultant from the OBO founder who helps us with ontology questions. And our friend Dennis Stevenson and his postdoc, Brandon Sin, from New York Botanic Garden, who was also involved in the plant ontology project, has been a really great deal of help. And of course, Georgios Goukos, who's a phenotyping expert in ontologies, and John Doonan, who is a high-throughput phenotyping person from Aberystwyth in Wales. And I also should acknowledge the group from Santa Barbara, B.S. Manjunath, who works on the BIS project. And a really important part of the Planteome is all the users and the resource databases who've contributed data and who use it on a daily basis. So many thanks to them, and many thanks to NSF again. So that's all.